Okay, so in this video, I'm gonna go over how to prepare a bacteria smear. So a bacteria smear is just a thin film of bacteria that you place on a glass slide, okay? And then I'm gonna walk you through how you can stain that bacteria smear, okay? So in this video, I'm going to go over two types of simple stains, and they are really very simple compared to more advanced or complex stains. So they're known as the simple stain and the negative stain. The simple stain is a direct stain, and the negative stain is an indirect stain, and I'll go over why one is called direct and why the other is called indirect, okay? But let me pose this question to you first. Why would it be important to stain cells, right? So most cells are colorless. All bacterial cells, all prokaryotic cells are colorless. Eukaryotic cells, the majority of eukaryotic cells are colorless with the exception of red blood cells, right? Because remember, what makes up cells? What did I tell you in lecture? So cells are mostly composed of water, and the remaining material are macromolecules. And those macromolecules really don't absorb any wavelength invisible light, okay? So therefore, they're not going to reflect color, okay? So why is staining important? So staining allows you to visualize that cell. It kind of allows that cell to come to life, if you will. So it allows you to look at the morphology. Remember, morphology is the shape, and then the shape, so remember the morphology is the shape, and then it also allows you to look at the arrangement of that particular shape. So if you remember, when we talked about bacterial shapes, there were three common bacterial shapes, right? So coccus, bacillus, and then spiral. So remember, for coccus, it's the spherically shaped bacteria, right? So it's a spherical shaped bacteria, but that spherical shape can appear as chains, and we call that strepto, remember? Or it could be a grape-like cluster, so there could be different arrangements of the shape, different arrangements of the morphology. Something else staining allows you to do, it allows you to look at cellular components. So you can look at cellular structures, right? So which type of cellular structures did we go over in lecture? Like the flagellum, right? The capsule, right? Pili, fibrilli, okay? Some other advanced staining allows you to look at the metabolic feature, or maybe it allows you to distinguish one cell type from another, which we will be doing with the Gram stain, which is a differential stain. These are not differential stains. They're not going to allow you to distinguish one cell type from another. It just really allows you to look at the morphology and the arrangement of a bacterial cell, okay? So what I want to do now is I want to, whenever I go over a simple stain and negative stain, I like to do a side-by-side comparison. So let's first talk about what makes the two stains similar, and then let's talk about what makes it different, okay? So I have this portion of the, let's say it's a table, divided, but this portion is open to show you what's similar, right? So what is similar between a simple stain and a negative stain? Well, a simple stain and a negative stain both allow you to view the morphology and arrangement, okay? And then something else, that's why it's so simple, is that it only requires one dye. When we do the Gram stain is a differential stain, so it requires two or more dyes to distinguish between cell types. Okay, so that is what makes both of them similar. So what makes them different? The difference really comes with the fact that for simple stains, they use positively charged dyes, okay? And negative stains use negatively charged dyes. So along with this video, you have background information, because it's important to read, right? On what makes the dyes positive and then what makes the dyes negative. Because I just don't believe in just talking about staining, how cells get stained and students don't understand why, okay? So the background information is going to tell you why it's positive, why it's negative, okay? So for simple stains, they use these positively charged dyes that can actually enter the bacterial cell. Why or how? The cell, right, so cells for the most part have a negative charge due to proteins. So the surface of bacterial cells in the cytoplasm is negatively charged. So we know that opposites attract. So that allows the stain, the dye, to penetrate the cell, okay? How does this compare to a negative stain? For a negative stain, because it uses negatively charged dyes, and remember the surface of the bacteria and the interior is negatively charged, gets repelled, right? So the negatively charged dye gets repelled by the negatively charged surface and interior. So the stain is going to remain outside of the cell, okay? So the stain remains outside. So basically you have a colorless cell against a stained background, okay? So what are the benefits? What are the pros and cons, let's say, for each type of stain? So the pro for a simple stain is that it allows you to see a stained cell, right? Because the dye can enter the cell. A drawback could be, let's say that you want to stain a cell, but you want to know the size of the cell. You want the accurate cell size. If you use the simple stain, you're not going to really be able to determine the accurate cell size, because if the stain can enter the cell, it's going to cause the cell to become a little bit bigger, right? So it's going to distort its cell size. Same thing for simple stain. Something else for simple stain is that there's a technique that's involved called heat fixing. Heat fixing helps to adhere the bacterial cells to the slide, especially if you want to preserve the slide. And heat fixing, because you're applying heat, can distort the cell size, okay? And heat fixing also, and I'll go over this again, heat fixing actually kills the cells. But you don't want to overheat fix, because you don't want to lyse or rupture the cells. You want the cells to be intact, or it's going to defeat the purpose of staining. For the negative stain, let's say the pro is that it can provide accurate cell size, because the stain doesn't enter the cell and there's no heat fixing, okay? So there's no heat fixing. So it's nothing there to distort, change the cell size. What's another pro is that there are heat-sensitive bacteria. So this is a great stain if you're working with a heat-sensitive bacteria, because you're not applying any heat. You're not doing any heat fixing. Okay? So I'm hoping that that was clear and give you a clearer picture of these two types of stains, and then how they compare to one another, and what's your goal in when you use a simple stain versus when you use a negative stain. Okay? Now I'm going to walk you through the simple stain first, okay, and then that's going to be followed by the negative stain. Okay, so now I'm going to walk you through the steps of the simple stain, okay? So the first thing you do, of course, is you're going to obtain your glass slide. You're going to wash the glass slide, and you're washing the glass slide to remove any type of debris or fingerprints, okay? So you wash your glass slide. The next step is that you're going to place your glass slide on a flat surface, or on that, I'm using a staining tray. There's a platform in which you can lay the glass slide, but you want to make sure you lay it on a flat surface. And then you're going to add a droplet of distilled water, okay? And you're going to add the droplet of distilled water at the edge of the slide, okay? And there's a reason why I'm having you this is kind of like what you teach to beginners when they are creating their bacteria smear. If you're someone that has been doing this for many, many years, you wouldn't approach it this way, okay? So you add your droplet of water, and it's distilled water, to the edge of the slide. And the reason why we are adding water is because we're taking bacteria that is growing, or that has been grown on a solid media, and that was agar. If we were taking the bacteria from nutrient broth, let's say, a liquid media, we would not have to introduce water, okay? So here is our droplet of water. The next step is that we add our bacteria, okay, to that droplet of water. But we have to apply aseptic techniques when we transfer that bacteria into the droplet of water, right? So we sterilize our inoculation loop prior to adding the bacteria, okay? And then once we add the bacteria, what you're going to do is that you're going to use the inoculation loop to mix the bacteria in with the water. And you want to mix it until it becomes cloudy. This is so very important, because cloudiness tells you that you have collected enough bacteria cells. So let's say after a few mixes or after a few seconds, you notice your mixture is not becoming cloudy, then you're going to have to go and transfer in more bacteria. Okay? So again, I'm mixing until it becomes cloudy. So I say that this is my inoculation loop. This marker is my inoculation loop. I'm going to mix this until it becomes cloudy. Okay? The next step, so step number 4, is now you are ready to create your bacteria smear. So say that this is your inoculation loop. This marker is your inoculation loop. So after you're done mixing, what you're going to do is you're going to lift up the inoculation loop. And what you're going to do is that you're going to start making a spiral or these semicircles, and you're going to gradually make it bigger. This is going to help to thin out or spread out your bacteria smear. Okay? So this is your bacteria smear. Again, if you're someone that's been doing this for many years, what you're probably going to do is take your bacteria, mix in some water, and that's your bacteria smear. But I want to show it from this angle here so that we can make sure that our bacteria smear is thin enough. Because when it's thin, when it's spread out, you don't have to wait forever for it to air dry, and also the stain can properly penetrate the cells. The worst thing to have happen is if you do all of this, you look under the microscope and your cells are colorless. There's no color, okay, because you did not allow for a thinner, bacteria smear to really allow the stain, the dye to penetrate. Okay, so you create your smear. This is how you create it. The next step is that you have to allow your smear to air dry. How will you know if it's dried? So when it goes from wet to dry, okay? So that time can vary depending on how you made your bacteria smear. After that, step number 6, you're going to take your slide, you're going to pass it through a flame. This is known as heat fixing, okay? You can also do alcohol fixation as well, but heat fixing is just much easier. So heat fixing, what happens is, you're passing your smear through the flame, but the heat fixing, what it does is that it coagulates. Another word, clumps, but we're going to use a scientific word, coagulates the bacteria proteins. And this allows the bacterial cells to adhere to the slide, okay? So this allows the cells to adhere, you want to say stick, to the slide, okay? And you don't want to overheat fix. And also, so let me go back. So heat fixing, of course, allows for adhesion, right? But also heat fixing kills the bacterial cells as well, okay? But you don't want to overheat fix because, yes, you're killing the bacterial cells, but you don't want to distort or rupture the cells. You don't want to lyse the cells, right? Because that's going to defeat the purpose of staining cells, because you want the cells to remain intact, okay? So after you heat fix, then you're going to begin applying your first reagent, your dye, basically your only dye, methylene blue. So this is how very simple this staining procedure is, because it only requires one dye. And there are several types of positively charged dyes you can use. I'm just going to have you use, or if you were in person, use methylene blue. Okay? So you're going to apply methylene blue, you're going to flood it, be very generous in the amount of methylene blue that you add or introduce onto the bacteria smear. And usually you can leave it on for 30 to 60 seconds. The good thing is that it's not so time sensitive, so if you leave it on longer, it's not going to do anything to hurt the bacterial cells. Okay? So after, let's say 60 seconds, then, I should have put this here, you're going to wash the slide. Okay? So what you're going to do when you wash the slide, right? You're going to angle the slide when you wash it so that the excess water rinses off the slide, okay? So again, after you apply the methylene blue, then you're going to wash the slide or rinse the slide with distilled water. And as you are doing this, which I will show you, you're going to angle the slide when you rinse it, okay? And what you should see after you're done rinsing and blotting around the bacteria smear. And what I mean by blotting is taking, you could take bibulous paper or lens paper and blot around the smear, not on the smear because you don't want to disrupt it, so around the smear to remove any excess moisture. And after all of this, right, you should see your smear, and then the last step would be to observe the smear under the microscope, right? And you would start off with 4 times, 10 times, and then go to 40, and then you would end at 100 times. With the 100 times objective lens, which is also known as the oil immersion lens. Okay, so I'm hoping that this was clear, right? Going through the steps of the simple stain. So now we're going to finally actually conduct the stain. I'm going to show you how it's conducted. Perfect. So now what we're going to do is I'm going to walk you through how you would prepare the smear and conduct a simple stain. So before you even start any type of work with microorganism or just research in general, you should always disinfect your work area. You disinfect your work area, and I told you my disinfection of choice is 70% ethanol, and I explained to you the reason why we use 70 compared to like 100 or a higher percentage, I should say. So we disinfect our work area before and after, we are conducting a technique or doing any type of experimentation. And then we also make sure we have the proper personal protective equipment. So I have my lab gown on, I have my goggles, which I also disinfected with 70% ethanol, and then I have my gloves, okay? So what type of tools, what do we need in order to conduct the simple stain? Well, of course, we need our bacteria. So this is the bacteria Micrococcus luteus. It's a spherically shaped bacteria, so it's cocci, okay? We need our stain. So the stain that we're going to use, and you have the option of using several types of positively charged stains or dyes. For this one, I'm going to use methylene blue, okay? We also need the inoculation loop. So if you saw the aseptic video that I have, I told you which type of inoculation tools you can use to transfer bacteria. So this is our inoculation. Remember, it has the wire and this is the loop, but we have to sterilize it prior to transferring and after we transfer. We have our staining tray. It's just easier to keep everything less messy and more organized. We have our glass slides here, a platform to place the slides. Here's our slides. And also we have distilled water. So if we were in person and I gave you, a bottle or a tube or anything like that, you always make sure that you read the labels, okay? So here it says distilled water. This is 70% ethanol here, because we don't want to use 70% ethanol in this simple stain, right? We only use it to disinfect. Okay, so I'm gonna push this over here, right? So that's kind of one good thing to do once you disinfect your area. Place that disinfectant far away from what you're doing, okay? And then we have a clothespin just to make handling of the slide much easier, okay? So let's get started. The first thing you would do is that you would wash or rinse the slide. So even if you take the slide from a nice fresh package, you always want to make sure you wash the slide to remove any type of debris or if there's fingerprints and things of that sort. So I'm going to take this. And I don't like to wash with tap water. Some do with tap water. I just like to wash with distilled water. And I went over in the video why it's called distilled, what makes it different compared to tap water. Okay. Next, what I'm going to do is, remember, there's different types of media, right? There's media that's solid, there's media that's liquid. So remember, we're taking this bacteria, because you can see the bacteria growth. For this particular bacteria, the colonies grow yellow, the color's yellow. So the bacteria is growing on a solid surface, which is the agar. So since it's growing on a solid surface, we have to introduce water to mix the bacteria, okay? So what I do is I add try to make it small, but it's okay if it's not small, a drop of water towards the end of the slide. I don't do it in the middle. Okay, and there's a reason why, and I'll mention that. I'll go over that in a moment. I make I do the small droplet of water at the edge of the slide. Once I do that, now I'm ready to sterilize the inoculation loop, okay? So remember, this is not the most perfect flame because you should see 2 cones, but either way, I sterilize my inoculation loop. And what do you want to do? You want to see that it glows, right? So it turns orange. And you not only want to sterilize the loop, you have to sterilize the wire as well, right? Because we're trying to prevent contamination, we're trying to prevent cross-contamination, okay? Once you sterilize the inoculation loop, you have to allow it to cool. So we don't wave it around, we don't blow on it, right? Because that defeats the purpose of trying to prevent contamination. So we allow it to cool, usually for 30 seconds or so, okay? But let's say if you are a little bit impatient, what you can do is you can take the loop portion and place it onto an area of the agar where there's no bacteria to allow it to cool. Okay, so that's what I'm doing now to allow it to cool. Now I'm going to be ready to get the bacteria. Okay, so I know that we're very visual people, right? You don't have to see clumps and clumps of bacteria. You don't want that to, you don't want to collect clumps because that can make your bacteria smear very thick. And we want to make sure that our bacteria smear is thin. And the reason why you want the smear to be thin is that it allows for adequate drying time, and most importantly, it allows for the stain to enter, penetrate the cells. Okay, so what I'm going to do now is that I'm going to take the bacteria and I'm going to mix it in with the water. And I'm going to mix it until it becomes cloudy. Why do you mix it until it becomes cloudy? Cloudiness tells you that you collected enough bacterial cells. Okay, so we keep mixing and mixing until it becomes cloudy. If you see that your mixture, so like here, is becoming cloudy, it's going to be hard for you to see. If it doesn't come cloudy after a few seconds, then you need to go back and get more bacteria. Okay, so I'm not sure, you probably can't see this, but you know what cloudiness looks like if you have water and you introduce something into the water until it becomes cloudy. So here it's cloudy. What I want to do now is, now I want to create my bacteria smear. Okay, and what you do to create the bacteria smear is that I lift up the loop. I don't drag it. I lift up the loop, and I'm going to mix it one more time, but I lift up the loop, and then I go into the middle of the slide, and I start making these small spirals, and then the spirals become bigger. So they're like small semicircles, and then the circles become bigger, okay? Once I make my smear in the middle, then I'm going to re-sterilize the inoculation loop. For some microbiologists, right, they probably would not do, like for me, I don't go and create my smear like that. Once you become very, very, advanced and used to doing this, usually you just take the bacteria and introduce it into the middle, and if you need water, you mix the water in. But for beginners, I like to show this method of creating the smear. Okay, so you start off with these small semicircles and you gradually make this circle bigger so that the bacteria smear thins out. Once I'm done re-sterilizing the inoculation loop, I can lay it back down. Okay, so after I have created my smear, now the next step is that I have to allow it to air dry. Okay, so once I allow it to air dry, then I'm going to heat fix. So how do you know when your bacteria smear is dry? Well, when you first created your smear, it looked wet. So when it looks dry, you know that it has been properly air dried. Okay, and I'm not sure if you can see that. Okay, but it's almost there. And I'm not looking at this, I'm looking at this. This is the smear. Okay, so I'm allowing it to air dry. Okay, once it's air dried, then I'm going to be ready to go. I'm going to try to go this way. I'm going to heat fix. So what heat fixing does is that heat fixing clumps, but the scientific word is coagulate the bacterial proteins, and it allows the cells to stick, to adhere to the slide. This is very, very important in staining, because the worst thing to have done is if you start adding your reagents and your cells start sliding off and there's nothing to stain. So this allows for the preservation of the bacteria onto the slide so that when you start adding the reagents, your bacterial cells are still there. Nothing has slid off, okay? So now what I'm gonna do is take this slide, I'm gonna pass it through the flame just a couple of times. So here, and I'll do it one more time, and that's it. You don't want it to linger in there. You don't want to pass it through the flame multiple times, because that can end up lysing, rupturing the cells. And the goal of heat fixing is to allow for adhesion, to allow for the cells to adhere to the slide, but also to kill the bacterial cells as well without compromising their shape. Okay, you want the cells to be intact so that you can stain them and look at the cellular components or structures of the cell. Okay, so once I'm done with heat fixing, next I am ready to begin the stain. This is how easy these staining, more simple stains are. So I'm using the dye, the stained methylene blue. All I'm going to do is flood it. I like to use the word flood. I'd be very generous with the amount of dye that I'm adding. For this particular type of stain, the time that the stain stays on is not so time sensitive. When you get to the gram stain, the differential stain, there could be some time sensitive reagents. But for this one, let's say we can leave it on there for 30 to 60 seconds, okay? So I like to leave it on for about 60 seconds just to give the stain enough time to penetrate the cells, okay? For the sake of time, I'm just gonna continue here. So once the 60 seconds, or if you leave it on for 30 seconds, the time is up, what you're gonna do next is that you're gonna wash or rinse the slide. And you're gonna wash or rinse with distilled water, okay? The best way to do this, and usually I just do it with my hand, or you can use the clothespin, is that you angle it here, and then you do your rinse. And you're gonna get a sigh of relief when you see your smear is still there, and it's also stained. Okay, so you see the smear, might look crazy, but this is the smear, looks blue. Okay, it's still there, so I did whatever I needed to do correctly, right? So I did the heat fixing part correctly to allow the cells to adhere. Okay. Once you've done with your dyeing and the rinsing, then you can take bibulous paper or you can take lens paper. I personally like to use lens paper, and you blot around. Okay, so you blot around the actual smear. Okay, just to remove any excess moisture. Okay, and then once you've done that, then you're ready to observe your smear under the microscope. And which lens from the video on microscopes do you observe your specimen at first? Remember, it's the 4 times. So you start with the scanning lens first, so that you can first locate where your smear is, and it gives you a brief overview of your actual smear. Then you start going to the 10 times, 40 times, and then you should end on the oil immersion lens. And which power was the oil immersion lens? Remember, it's 100 times objective, and that's the one you can apply oil. That's the only objective lens you can apply oil to. And that'll allow you to get a real nice crisp image of your actual cells. So one thing that I can't do is that I wish I could show you the smear under the microscope, but I don't have a camera to attach to the microscope. So I'm just gonna show you some pictures of what the Micrococcus luteus should look like when it's stained with methylene blue. Okay, thank you. So now I'm gonna walk you through the steps of the negative stain, okay? So for the negative stain, you need 2 glass slides, right? So see how that compares to the simple stain. We just needed 1 slide, 1 glass slide. For the negative stain, we need 2 glass slides. Why do you need 2? Because one of those slides is going to be known as your spreader slide. And what it's going to do is that it's going to help you to spread the mixture across the first slide. So you have 2 slides. Slide number 1. You're going to, at the edge of, let's say, slide number 1, you're going to add your drop of water first, You're going to follow that using aseptic techniques and add the bacteria, okay? And remember, we have to still mix in the bacteria until it's cloudy, because cloudiness tells you that you collected enough bacterial cells. Once your mixture here is cloudy, then you're going to add in your dye, nigrosin, okay? So nigrosin is a black dye, okay? And once you add the dye, now you're going to be ready for your second slide. So this is the first slide, where all these three components go onto. And then you have your second slide, your second glass slide is a spreader slide. And what you're going to do is, say that this is your spreader slide. This is your spreader slide. You're going to take this here, this is your mixture here, and you're going to spread it across the first slide, okay? Once you do that, so this is very easy technique, staining technique. Once you've spread the mixture across the first slide, then you allow it to air dry. This is probably the most important part of this stain, because you don't want any areas, because you're spreading the mixture across, you don't want any areas of that first slide to be wet. And then you go and try to observe your cells under the microscope, because you can end up damaging the actual lenses, right? Because then the stains can get up into the lens. We don't want that to happen. So you really have to make sure that you properly air dry. Another trick is to make sure you add a small amount of nigrosin. If you add too much, that's definitely going to increase your air drying time. So the smaller the amount, the better. Okay? So again, after you let it air dry, then it's easy as that. There's no wash step, there's no heat fixing. Now you're ready to observe your cells under the microscope. And remember, what you are going to see is, say this is your cell, the bacterial cell, and you're going to see, and of course I'm going to have a better picture for you, an image, you're going to see that the dye, right, remains outside of the cell. So the cell itself will be colorless against a stained background. Okay? So now what I'm going to do is I'm going to perform the actual negative stain. So now we're going to do the negative stain, okay? So let's first talk about what you should do before you actually conduct the stain. You should disinfect your work area with 70% ethanol. You should make sure that you have the proper personal protective equipment. So I have my gloves, I have my goggles, which I also disinfected with 70% ethanol, and then I have my my lab gown, okay? So what material do we need to conduct the negative stain? We need our dye. This is our nigrosin. And I'm going to switch up the bacteria this time. We're going to use our good old Escherichia coli, known as E. coli. Okay. Also, inoculation loop. We need that to transfer the bacteria aseptically. I have the staining tray. We have our two glass slides that are on the platform. All right. And I also have the distilled water. Okay. So let's get started. So the first thing that I'm going to do is, I've already washed the slides. So I've already washed these slides just to save time. So what I'm going to do now is that I'm going to add, this is going to be considered our first slide. This is going to be the spreader slide. Okay? This is our first slide. This is the spreader slide. So onto the first slide, I'm going to add a drop of water. Okay? And after I add a drop of water, and you might want to pause the video each time so that you can start trying to remember, you know, the steps that follow as well, okay? So what happens after you add the drop of water? Next, you're going to aseptically transfer the bacteria into the droplet of water. So I have to sterilize the inoculation loop, right? And proper sterilization is when you see that it turns, the wire in the loop turn orange, so it looks like it's glowing. Okay, so it's not about just sterilizing the loop itself, you have to sterilize the wire as well. Okay, so once I do that, I allow the inoculation loop to cool, remember, for about 30 seconds or so. But just to kind of, for the sake of time, what I'm going to do is, I'm going to, this is the bacteria, this is the E. coli, I'm going to bring the loop onto an area of the agar where there's no bacteria, okay, to allow it to cool, okay? And sometimes you would hear it sizzle. Sizzling is bad. if you go directly onto the bacterial cells, because that means that you are rupturing the cells. So now once I allowed it to cool, I'm going to collect the bacteria. Okay? And also, as soon as you're done collecting the bacteria, you want to make sure you close the Petri dish right away. So now I'm going to introduce the bacteria into the water. I'm going to mix until it becomes cloudy. Because remember, what does cloudiness tell you? Cloudiness tells you that you collected enough bacterial cells. And this is perfectly cloudy. So I'm going to re-sterilize the inoculation loop. And you can see I have a clump of bacteria here, so I want to make sure I definitely get that off. And again, this is not the most perfect flame here because you should see the 2 cones, but you were introduced to that in a previous video. Okay, so then now I can place this inoculation loop down, and now I'm ready to add the, hopefully it's going to be a small drop of omnigrosin. Okay, so right here, perfect. Okay, right onto the mixture. So you cannot do it in the wrong order. You can't just go and add nigrosin, then the water and the bacteria, because then if you do it that way, you won't know if you collected enough bacterial cells. Okay, so now what I'm going to do is I'm going to take this slide here, okay, and then I'm going to actually rearrange it this way. And I'm going to take this slide here, and then I'm going to spread it here. So I usually do it at an angle, so it's already starting to go, but that's okay. I'm going to spread it across like that. You might have to do it a few times, but that's okay. Some like to do it with a clothespin. They think it's easier. I just like to do it like this. And once I spread it across, I lay it flat. What I'll do with this one is that I will discard it into a biohazard bag there. And then what you're going to do next is be patient, unless you have something else to do, while you're waiting for this to dry. So this needs to properly air dry. So once it properly air dries, you can tell when it's dry because it's going to look dry. Then you are ready to observe it under the microscope. There's no heat fixing. There's no using lens paper or bibulous paper to blot around. You leave it as is, and then you're ready to observe it under the microscope. Let's say that you go from the 4x10 and then to the 40 and then to the 100 times objective lens where you have to apply oil. You would not apply oil to the whole slide. You would just apply oil to a portion of the slide, and it would be a very, very small drop of oil, because remember, when the 100 times objective lens comes into contact with the oil, it's going to spread anyway. So you want to make sure that only to a small portion of this slide, you add a small droplet of the oil. You don't have to add oil. You should not add oil to the whole slide. Okay? Thank you.